right, everyone. Okay, we're gonna get started. Couple quick reminders. We do have two things this week. So we're gonna have lecture today. We're gonna have lecture on Wednesday. After Wednesday, you do have your next online Brightspace quiz that will open up after class on Wednesday, and it goes through to Thursday night at midnight. You do also have two sets of interactive notes due by Friday. I realize Friday is also Good Friday. Good Friday means we don't have class this Friday, and then we have Easter weekend. I am going to keep the interactive notes Dropbox time the same. I know it's Friday at 5 p.m. That's Good Friday. If you don't want to have to work on Good Friday, because it's when we're supposed to have it off, just get them in earlier, okay? But I will keep the deadline kind of just the normal deadline, even though it is Good Friday, okay? Don't expect you to do it on that one if you don't want to work on Friday because we don't have class that day, but do get it in early if that's the case, all right? So that is what we have for this particular week. We'll also be lecturing then next week. And remember, the third exam is coming up on April 17th then. Okay? All right. All right, last lecture, we left off talking about citric acid cycle, about respiration. Not cellular respiration, just respiration by itself. When I say the term respiration, what am I talking about? What process is happening or what is being used when I say the term respiration? Respiration is defined as what? Use of an electron transport chain. Yeah, to make a proton motive force. So remember, with a respiration, this is where electron transport chains is a series of enzymes. Where are those enzymes located? In prokaryotes. Where are the electron transport chain enzymes located in a prokaryotic cell? In the plasma membrane. So remember, respiration, this is going to be, again, use of an electron transport chain. These are a series of proteins in the plasma membrane, okay, to make proton motive force, a proton gradient across a membrane. They pump protons, H+ ions, to one side of the biological plasma membrane. Okay. All right, so what ends up happening here is that this is going to create that proton motive force. It's also going to be, what is the typical source of electrons for respiration? What is typically often the source of electrons for respiration? Yeah, it's going to be the NADH. Okay, NADH stores high energy electrons. NADH is going to transfer those electrons from NADH into the electron transport chain. Those electrons are going to move from one member of the electron transport chain to the next, to the next, to the next, to the next. And the energy from those electrons is used to then pump protons across the membrane and make a proton motive force, which is one of the three main energy intermediates in there. What's going to be happening here with these catabolic reactions? Remember, one of the problems is that glycolysis is really efficient. And so all of the NAD+ in the cell can get converted into NADH by glycolysis. And then the cell runs out of NAD+, glycolysis can't keep running. So in catabolic reactions, typically glycolysis is either coupled to fermentation or respiration. And that NADH gets used up in both fermentation or respiration, but it gets used up in different ways. In fermentation, the NADH is used up, that regenerates NAD+, the NAD+ keeps glycolysis running. And in fermentation, the electrons from NADH are returned and put onto pyruvate, but then it makes a waste product the cell doesn't want. Something like lactic acid or ethanol. If those build up, it's actually bad for the cell. That's okay because it keeps glycolysis running, and glycolysis is processing energy for the cell. Sorry. In respiration, again, NADH is used as an electron source. NADH gets used up. That regenerates NAD+. The NAD+ is going to keep things like glycolysis and citric acid cycle running. But that NADH is used up much more productively. Instead of just wasting that NADH energy and making a waste product, the electrons, the energy from those electrons in NADH is used to then, by the electron transport chain, make a proton motor force, make another different energy intermediate for the cell. So in respiration, you use up the NADH, but you produce something useful for the cell off of it as well. Okay. Questions about that? Those three energy intermediates, I brought them up quite a bit, but all cells need the three energy intermediates, pretty much all cellular life. Whether we're talking about a prokaryote, a eukaryote, a plant, an animal, a fungus, a bacteria, all cellular life uses those three main energy intermediates of NADH, proton motor force, and ATP. Those are the big three. Okay? And so if I say an organism, for instance, is respiring glucose, it's only two words. But if I say something is respiring glucose, what that means is glucose is going in. But what pathway does glucose always go into? Does it go first right into respiration? No. It first goes into glycolysis. So glucose goes in, it goes into glycolysis. That glycolysis, what's glycolysis going to make? Two net ATPs, two NADHs, and two pyruvates. If it is doing respiration, citric acid cycle and respiration always go hand in hand. If they use one, they're using also the other. So species that are doing respiration also always have citric acid cycle. Okay, what ends up happening then is my pyruvates are going to go into the citric acid cycle. The citric acid cycle is then going to break those pyruvates down. It makes even more NADH. How much NADH per a pyruvate? I think somebody said it. Makes 5 NADH equivalents. It makes 1 ATP equivalent per a pyruvate that goes into that then. So that citric acid cycle is going to be making an ATP equivalent, but it actually makes quite a bit of more NADH equivalents then as well. Those NADHs, both the NADHs from glycolysis, those NADHs from then citric acid cycle, those then feed into the electron transport chain. The electron transport chain is going to make is going to be that process of respiration. That then makes a proton motor force. In essence, I'm converting the NADH in one of the three energy intermediates into a different energy intermediate. That proton motor force can sometimes get used directly, but sometimes that proton motor force can also get converted by the action of ATP synthetase into ATP. So ATP synthetase is an enzyme complex that makes ATP, but it needs an energy source to do that, and the proton motor force is the energy source for ATP production. By the way, where's ATP synthetase located? Where did I show it located when we went through slides? Yeah, it's also in the plasma membrane. So electron transport chains are in the plasma membrane. ATP synthetase is in the plasma membrane. Glycolysis, citric acid cycle are both in the cytoplasm. Okay. That's in prokaryotes. It's a little different in eukaryotes. Now, I could also say something like an organism is then fermenting glucose. What this then means, this description is going to basically mean it's doing, if glucose is going in, what process is it always doing? Yeah, so glucose is going to go into glycolysis first. But if I say it is fermenting glucose, that means glycolysis is getting coupled with fermentation instead of respiration. So we're talking about the processes here being glycolysis plus fermentation. Glycolysis. What's the products of glycolysis again? 2 net ATPs, 2 NADHs. So when that glucose goes into glycolysis, you are getting some NADH production. You're getting some ATP production. And that's, you're getting, again, 2 net ATPs, 2 NADHs, 2 pyruvates is what comes out of glycolysis. But I also just said that all cells need the 3 energy intermediates. All cells need to make NADH. All cells need to make ATP. Also, all cells do need to make proton motor force, including, for instance, our cells. Okay. Does glycolysis make proton motor force? No. Does fermentation make a proton motor force? No. And there are some bacteria, their metabolic strategy is only glycolysis coupled to fermentation. That's it. That's only their metabolic strategy. That's all they ever do. Glycolysis, fermentation. There is no respiration. There's no electron transport chains. There's none of that. But they still do need a way to make a proton motor force, even though a lot of fermenting bacteria do not possess an electron transport chain. So they have an alternative strategy for proton motor force production, and it is something called reverse ATP synthetase. This is in fermenting bacteria. Okay. So reverse ATP synthetase is similar to ATP synthetase, but it is a different enzyme. Okay. And it does, in essence, the reverse of ATP synthetase. So remember, during respiration, you have a ATP synthetase, not the reverse one. You've got the ATP synthetase. Okay? And in this case, what happens is proton motive force is an energy source for ATP synthesis, making ATP. So this is going to be the action of ATP synthetase during some processes like cellular respiration, for instance. Okay. In fermentation, what they have instead is this reverse ATP synthetase. And what it does, it actually uses ATP as an energy source to pump protons and make proton motive force. So fermenting organisms, specifically fermenting organisms, they have this reverse ATP synthetase enzyme. This consumes the ATP, uses it as an energy source to make the proton motive force and pump protons across the membrane. Questions about that? Again, I want to differentiate a little bit when I say respiration versus cellular respiration. When I say just the term respiration by itself, what it tells you is that electrons are getting donated to an electron transport chain. The electron transport chain is using the energy from those electrons to then make a proton motive force. Okay. By the way, that term cellular respiration that you guys might have had before or gone through in a class like NU 120. Cellular respiration is actually a combo of metabolic pathways. So when we say the term cellular respiration instead, this is actually a series of connected metabolic pathways, but it actually, cellular respiration as a whole, it involves glycolysis plus citric acid cycle plus respiration, which is the electron transport chains making a proton motive force, plus also a process called chemiosmosis, which is actually ATP synthetase making ATP. Okay, so when we say that term cellular respiration, it's a catch-all for all four of those things added together. Four different metabolic pathways added together. They go all the way from glucose, glycolysis, citric acid cycle, to respiration, to what's called chemiosmosis, which is then using ATP synthetase. Questions about that? Okay. Respiration by itself is just this part. When I say the term respiration by itself, it's just that one part. Cellular respiration is all of those added together. Okay. Now, when I say also respiration, it means electrons are donated to an electron transport chain and a proton motive force is being made. So that proton motive force is being made. I said that a common electron donor is NADH. It's not the only electron donor. Electron transport chains, especially bacterial electron transport chains, are actually flexible systems. You guys ever heard the phrase, don't put all your eggs in one basket? What's that mean? Don't put all your eggs in one basket. What's the problem if you put all the eggs in one basket? What's the problem with that? Say you trip and fall, all your eggs break at once. Okay, it means diversify. It means don't put everything all in one spot. Okay, diversify so you're less likely to have an accident mess things up, for instance. Well, biological organisms do need to diversify as well. You don't want to count on only NADH. You don't want to put all your eggs into the NADH basket, because if you run out of NADH for some reason, the cell is going to be in trouble. So it turns out you can have different electron donors. So NADH is a very common electron donor to electron transport chains. It's not the only one. I'm simplifying things when I teach it that way. Okay, and we're not going to get into it too much in detail, but you can have different electron donors putting electrons in different places. So for instance, something like methanol, CH3OH, that can also be an electron donor to electron transport chains. You can have different ones. You can have NADH putting electrons in. You can have something like a chemical like succinate can act as an electron donor to this electron transport chain. You can have methanol putting in electrons into the electron transport chain. NADH is probably the most common electron donor to electron transport chains. It's not the only one. Okay, you don't want to put all your eggs in one basket type of situation here. So electrons from different chemicals can enter the chains at different levels of the electron transport chain. NADH is usually going to put its electrons in the very first member of the electron transport chain. Something like methanol, it puts electrons in towards like the end of the electron transport chain. And you get different amount of protons pumped. So if you have electrons come here at the beginning, you pump protons here, you pump protons here, you pump protons here. So something like NADH, you're pumping 4 protons here, 4 here, 2 here. You've got like 10 protons pumped with something like NADH. This has really high energy electrons on it. Something like methanol, it has low energy electrons. And so they come in later and maybe you only get 2 protons pumped. Okay? It has lower energy electrons. You still get some protons pumped, but less. So you're getting some different yields of energy. NADH is probably one of the more common electron donors because it has really high energy electrons and you get a lot of protons pumped off of it. Other electron donors, you get other amounts. Questions about that? The other thing is these are flexible pathways. It's also flexible in the exit. So respiration can also have different what's called electron acceptors, or sometimes these are referred to as terminal electron acceptors. So what accepts the electrons at the end of the electron transport chain? In aerobic respiration, which is basically cellular respiration, electrons get passed through an electron transport chain. Electron transport chain makes the proton motive force, but then they get passed to oxygen as the terminal electron acceptor. This is the case in what is called aerobic respiration. Aerobic respiration refers to oxygen being used as a terminal electron acceptor at the end of the electron transport chain. Many species of bacteria are going to do aerobic respiration. But there are other species of bacteria that instead do something called anaerobic respiration. Anaerobic respiration, this is where electrons are donated and passed through an electron transport chain. That electron transport chain is still going to make a proton motive force because that's what electron transport chains make every single time. Electron transport chains always make proton motive force. But then in anaerobic respiration, some molecule other than oxygen, this is going to be your terminal electron acceptor at the end of the chain. In anaerobic respiration, something other than oxygen is going to act as a terminal electron acceptor. Now, what molecules do I mean by other than oxygen? There's quite a few. Electrons. In anaerobic respiration, electrons go into electron transport chain. The energy from those electrons are passed through the electron transport chain and used to make a proton motive force. But at the end, those electrons have to go someplace at the end. And they're going to go onto a molecule other than oxygen. Some common examples of this might be certain species might use NO3- nitrate as a terminal electron acceptor. Some other species might use then selenium as or carbon dioxide or iron or fumarate. So these are some examples of molecules used as terminal electron acceptors. But as long as it's something other than oxygen, we consider it a type of anaerobic respiration. Questions about that? Okay. Just as an example of this, there's a bacteria called Paracoccus denitrificans. It uses nitrogen compounds like nitrate, nitrite, nitrous oxide, nitrogen gas as terminal electron acceptors. So NADH is going to be its electron donor. NADH will put electrons into the electron transport chain. Those electrons will pass through the members of the electron transport chain, but then they go onto these nitrogen compounds at the end, and eventually they make as a waste product nitrogen gas. And they just release that nitrogen gas out into the atmosphere. Questions about that? All right, what I'm just pointing out here is electron transport chains, especially in bacteria. Electron transport chains are very flexible. They can have different electron donors. So you can have these electron donors to an electron transport chain. Then you can have different possible electron acceptors at the end of the electron transport chain. So this is going to be the start. This is going to be the end. But that electron transport chain, no matter what it uses as a donor, no matter what it uses as an acceptor, it's making proton motive force. The other thing I want to get into as we go through stuff today is that different types of bacteria do different types of metabolism. So for instance, mitochondria. What's the function of mitochondria? Mitochondria is an organelle in our cells. What's its job? It creates ATP and it is energy processing. It does not create energy. Remember, it processes energy. I just want to actually point that out. Energy, first law of thermodynamics is there is a limited finite amount of energy in the universe. Okay? There is X amount of energy and there's always X amount of energy. Energy cannot be created or destroyed. Organisms do not create energy. They harvest and process energy. And so what a mitochondria is doing is it's processing energy and converting or transforming that energy into ATP, chemical energy. But it's taking one energy source and using it to make ATP, which is the way the cell stores energy. Okay? Mitochondria has what we call an optimal electron transport chain. Its electron transport chain is really good. And by doing something like cellular respiration, it is involved in cellular respiration, but then by doing glycolysis coupled to citric acid cycle, coupled to respiration, coupled to chemiosmosis, all four of those processes that I listed out earlier, it makes about 36 ATP per a glucose molecule that goes in originally. Okay, so if you start off with one glucose, you end up getting about the equivalent of 36 ATP molecules being made off of a single glucose. E. coli also does respiration, but its respiration chain is not as good. Its electron transport chain has what we sometimes refer to as a suboptimal electron transport chain. It's still really good, but you end up only getting about 20 ATP per a glucose molecule that's processed. Streptococcus. This is a fermenter. Streptococcus is a fermenting bacteria and only a fermenting bacteria. It specializes in doing fermentation. That means it has glycolysis coupled to fermentation. That's it. And then you get 2 ATP per glucose in that fermenting bacteria. Where do those 2 ATP come from? What process makes those 2 ATP? Glycolysis and just glycolysis. Remember, it's glucose goes in to glycolysis. You're getting 2 net ATP, 2 NADHs, 2 pyruvates out. That's where it's getting its energy from. Okay? In this particular case, if we're going between E. coli or Streptococcus, which one of those is most efficient? That is going to be E. coli. The reason is efficiency is a description of how much energy an organism can extract from a source. And the more energy per source, the more efficient. Questions about that? Okay. Because we're getting more AT per glucose, it's more efficient. Between E. coli and Streptococcus, which one do you think grows faster? Well, we said E. coli is more efficient, so you might think it grows faster. It doesn't. Turns out the faster growing species is actually Streptococcus. In fact, in many cases, growth rate, in some cases, not all, in some cases, growth rate has nothing to do with the efficiency, strangely enough. And in many cases, the fastest growing bacteria are the fermenting bacteria. However, most fermenting bacteria have evolved in what is called nutrient-rich environments or eutrophic environments. That's a term we've had previously. Fermenting bacteria have typically evolved in very high nutrient conditions, so they don't have to worry about nutrients. They don't have to worry about being efficient, in essence. And in fact, they're just trying to consume those nutrients as fast as possible and grow as fast as possible. Also, fermenting bacteria are simpler. They have less parts to make. Fermenting bacteria don't make electron transport chains. They don't make citric acid cycles. They don't make a lot of stuff. And they're hyper specialized into just doing glycolysis and fermentation. They do it as fast as they can, processing as much sugars as they can, as quick as they can. And that lets them grow really quickly, then, even though they're not efficient. They do tend to burn through their nutrients very fast, though. They typically, sudden influx of nutrients, they grow real fast, then they run out of nutrients and their population almost instantly goes into like stationary phase or death phase. So they typically grow fast and then consume everything. What I'm also trying to hit here is that different bacterial species do different types of metabolic reactions. So for instance, some bacteria, they specialize in coupling glycolysis and respiration. And that's what they do. Glycolysis coupled to respiration. And so for instance, many species that are obligate aerobes. What does that mean? What are obligate aerobes again? They grow only where? In O2 conditions. So they only grow if oxygen is around. And what they do is they specialize in aerobic respiration. Some examples of this are things like Pseudomonas aeruginosa, Mycobacterium tuberculosis, a couple of the pathogens of interest that we talked about previously. Where does Mycobacterium tuberculosis cause infections in our body? In our lungs. Guess what's a lot of, what is there a lot of in our lungs? Oxygen. Okay, it turns out Mycobacterium tuberculosis is an obligate aerobe. It can only grow in the presence of oxygen. It actually needs oxygen for its aerobic respiration. So it uses oxygen gas as a terminal electron acceptor at the end of its electron transport chain. So it requires oxygen to be present for it to grow, in essence, then. Okay. Some bacteria specialize in doing glycolysis coupled to fermentation. This is going to be many species of obligate anaerobes or aerotolerant bacteria. If they fall into this obligate anaerobe category, if they fall into the aerotolerant category, they're typically doing glycolysis coupled to fermentation, and these are fermenters. Some examples of this is like Clostridium species, Bacillus anthracis, which causes anthrax falls in this case, Clostridium difficile, which causes C. diff infections, for instance, falls into this particular category as well. Again, this is going to be that these guys only grow in the absence of O2. These grow independent of O2 concentration. That means they grow equally well in high or low O2. Okay. But these are going to be fermenters. Now, there are still some other bacterial species that can perform glycolysis, fermentation, and respiration. So what they're going to do is sometimes they're going to be glycolysis plus fermentation, but other times glycolysis plus respiration. They can switch back and forth. Okay, so sometimes glycolysis coupled to fermentation. Under other conditions is glycolysis coupled to respiration. And they can switch it up depending upon their environmental conditions. These are the facultative anaerobes. This includes species like Escherichia coli, Staphylococcus aureus or Staphylococcus epidermidis, Listeria monocytogenes. It includes things like Baker's yeast, Saccharomyces cerevisiae. It even includes us to a certain degree. We can do glycolysis, but we can also sometimes couple glycolysis to fermentation. Usually we have to couple glycolysis to respiration. Under what conditions do you think a facultative anaerobe would perform fermentation? When would it do fermentation? What environmental conditions? Yeah. When would it be doing respiration instead? With oxygen. So typically what happens is, remember, facultative anaerobes, they can grow both in the high oxygen and in no oxygen environments, but they prefer growing in oxygenated conditions. So if we remember with that definition of facultative anaerobes, we had that little tube, and I use that as this, but the facultative anaerobes, they can grow everywhere, relation to oxygen concentration. So this is going to be high O2 at the top and maybe no O2 at the bottom. But they prefer growing where the oxygen is. And you're going to see a higher density of them in the high oxygen concentrations. Does that make sense, everybody? Okay. Why would they prefer oxygen concentrations? Yeah, with O2, they make more ATP per a glucose. Remember, with fermentation, you're going to be getting about 2 ATP per glucose. Here, you're going to be getting anywhere from often 20 to 36 ATP per glucose. So they're going to be more efficient. They're going to get more ATP per glucose with respiration compared to fermentation. Questions about that? Oops, yeah. So again, are they going to grow more efficiently in the presence or absence? They're going to grow more efficiently in the presence of oxygen, because with oxygen, they do glycolysis coupled to respiration, and you get more ATP per a glucose then. Questions about that? All right, I've been talking a lot about glucose. One thing I just want to point out, though, that's a simplification. Glucose is a very common nutrient source. It's a very common thing that you use to start this process. It's not the only thing. It's not the only sugar that's going to be used. And sometimes you can also start with not sugars. Sometimes you can start these metabolic pathways with something like an amino acid instead, or a fat. Okay. Glucose is used as a simplification to go through and teach some of these processes. Turns out it's much more complicated than that, though. Okay. But that's a topic that's gone through in something like a upper level biochemistry course, where they're going to start to map out, oh, amino acids enter into these metabolic pathways here, or lipids enter in here, or glucose enters here, fructose enters in here. And so they start to talk about different molecules starting some of these processes and where they feed in, and they map that stuff out then, for instance. Okay. We're not going to go through that. We're just going through mostly glucose, but other things can be processed as an energy source. So for instance, say we're talking about lipid catabolism. Lipid catabolism, this means we're using a lipid that goes in, a fat goes in. And so for instance, this is what is called a triglyceride, or a fat molecule. This is a glycerol. Actually, this part in black here, the part in black here, this is a glycerol molecule. To that is attached 1, 2, 3 fatty acids. So this entire thing right here in red is 1 fatty acid. In blue is a 2nd fatty acid. In green is a 3rd fatty acid. And this is attached onto this glycerol molecule, which is this guy up there. Okay. And this is what is called a triglyceride, or a fat molecule. So when we're talking about fat molecules, this is what they look like chemically. What happens is this is actually a fairly nice energy-rich molecule, and the cell would like to break it down. This will go into lipid catabolism. Typically, the first thing is that phospholipase comes in and it cuts right here. It cuts one of those fatty acids off of the triglyceride. Now you've got a free fatty acid. And so that's what this guy is right here. This gets attached to something called coenzyme A. Where have we talked about coenzyme A before? What metabolic pathway was coenzyme A part of? What? It's attached to acetyl-CoA, and then it goes into what metabolic pathway? Citric acid cycle. So coenzyme A was also involved in the citric acid cycle. But now you got a fatty acid to coenzyme A. This is going to go into a pathway called the beta oxidation pathway. And the function of the beta oxidation pathway is called lipid or fatty acid catabolism. That's just my way of saying it breaks down fatty acids as an energy source for the cell. So when I say catabolic reactions, remember catabolic reactions are going to break large molecules down into smaller molecules and make energy intermediates. When I say lipid or fatty acid catabolism, it's going to break a fatty acid down into smaller molecules and it generates energy intermediates for the cell. So if I'm just adding on those together. In the beta oxidation pathway, what goes in is a fatty acid attached to coenzyme A that goes in to the beta oxidation pathway. Okay? What's going to happen is it goes through a series of chemical reactions, but you make a NADH, you make a FADH2, that is 2 NADH equivalents, and you make an acetyl-CoA. So this is going to be this acetyl-CoA, that is an output. These 2 NADH equivalents, that is also an output from this pathway. My input is that fatty acid attached to a coenzyme A. The NADH that's made can feed into electron transport chains that generates more energy for the cell in the form of proton motive force. The acetyl-CoA, this can go feed into the citric acid cycle, which then makes even more NADH off of it, which then also feeds into the electron transport chain, which makes some proton motive force for the cell. Okay, so these are all connected together. There's also a left, and so what ends up happening, say you have a 16 carbon fatty acid that goes in. What ends up happening is one time through the cycle, what you get is an acetyl-CoA. This is a 2 carbon acetyl group, but you also get a 14 carbon fatty acid left over. Turns out that 14 carbon fatty acid could just go right back into the beta oxidation pathway. The 14 carbon acid goes back through. You get 2 NADH equivalents to an acetyl-CoA, and you get a 12 carbon fatty acid. 12 carbon fatty acid just goes right back into the beta oxidation pathway. And what the beta oxidation pathway is, it tears apart fatty acids 2 carbons at a time. It breaks off 2 carbons in the form of acetyl-CoA, and then it generates more NADH then. Questions about that? So that's actually going to generate quite a bit of energy for the cell. All right. We've talked about different modules. And by modules, I'm talking about metabolic pathways. So metabolic pathways are going to be things like beta oxidation pathway or glycolysis, citric acid cycle, electron transport chains. Those have been metabolic pathways, and metabolic pathways are a series of interlinked chemical reactions. But for instance, glycolysis was a series of interlinked 10 chemical reactions through the process of glycolysis. There was a series of 10 chemical reactions, okay? For each metabolic pathway, like glycolysis, know the input, know the output for each pathway. What goes into glycolysis, what comes out of glycolysis. What goes into citric acid cycle, what comes out of citric acid cycle. What goes into fermentation, what comes out of fermentation. But also know connection points / molecules between pathways. So for instance, what connects together glycolysis and citric acid cycle? Acetyl-CoA, but even something before that. What molecule connects glycolysis to the citric acid cycle? Pyruvates. The pyruvates produced from glycolysis go into the citric acid cycle. What molecule connects citric acid cycle to electron transport chains? NADH. The NADH made from the citric acid cycle will then feed its electrons into the electron transport chain. So NADH is the connection molecule between them. Okay. So you should know the connection points between these as well. You should also know, so there's kind of like this part you need to know, this part you need to know. You also need to know locations. So I've asked you those one a few times, but for instance, where's glycolysis located in a prokaryotic cell? In the cytoplasm. Okay. Where's fermentation located in the prokaryotic cell? Also in the cytoplasm. Questions about that? Or questions about the metabolism unit then? All right, next lecture, we are switching gears a little bit. We're going to start talking now. We've kind of finished up our catabolic unit. We're going to start going into anabolism now, making macromolecules. Okay, so we're going to be, instead of like energy processing, we're going to be starting to use up the energy intermediates to make macromolecules. We're going to start off talking about genetics then, and making nucleic acids.